Hello, everyone. Thank you for joining us today. As a reminder, all GSA online events are covered by a code of conduct, and the link for that will be available shortly in the chat. Hugo, you are good to go.
All right. Welcome, everybody. Today, we're going to hear about MARRVEL, mining genetic and genomic data across model organisms. And the talk will be presented by two PIs from Baylor College of Medicine, Shinya Yamamoto and Zhandong Liu. First, a few words about Shinya. Shinya obtained a degree in veterinary medicine from the University of Tokyo and applied to the Baylor graduate program, was accepted as a graduate student and did exceptionally well. He then joined Baylor College of Medicine as a faculty and has been assistant professor for the last 4 or 5 years. So his interests are mainly to solve human genetic disease and participate through the Undiagnosed Diseases Network in the diagnosis of human diseases and work out the mechanisms as well. He also has some other interests which relate to dopaminergic neurons and the biology of dopaminergic neurons and nudge signaling. And today he'll talk about MARRVEL and what he, in collaboration with Zhandong Liu, have developed, which is a very useful tool to search a lot of data. The other speaker is Zhandong Liu, who did his Bachelor's of Science in Nankai University and then joined the University of Pennsylvania, where he did his PhD. He's a bioinformatician and is really using computational tools to study large biomedical data sets. He's an associate professor in the Department of Pediatrics and has a large team that really helps us develop tools to mine all these data. And he's currently working on artificial intelligence, which he'll talk about today, to try to really develop artificial intelligence to mine all these data and make predictions with respect to the genes that cause human disease when we don't know what variant is really responsible for a phenotype. So I'm going to pass the slides to Shinya. Shinya?
Okay, do you hear me, Hugo?
Yes.
Great. So my name is Shinya Yamamoto, and together with Zhandong, my colleague here, we're going to be telling you about MARRVEL, which is actually a tool that actually we've developed together with also Hugo and other folks in the context of the Undiagnosed Disease Network. Just want to see if my mouse is working here. Okay. So the overall, so the goal of today's meeting, the webinar, is to give you a background of why we developed MARRVEL in the first place. And this is 4 years ago in 2017. And then I'll do a live demo of MARRVEL so that you can actually see how we actually use this information that MARRVEL gathers. And then I'm going to be passing the baton to Zhandong, who will talk about how machines, computers, can actually also use this data to develop new useful tools for the community. And finally end up with question and answer sessions. Hopefully we'll have like 10 minutes for that so that we can answer any questions you have. If we don't fully answer your question, just don't hesitate to contact us via email or through GSA. So MARRVEL was, again, developed in the context of the Undiagnosed Disease Network. And we're actually, together with Hugo Bellin, Michael Wangler at Baylor College of Medicine, we function as the FlyCore of the Model Organism Screening Center. So to give you a kind of a little brief overall about the UDN, there are many patients that are have rare diseases that are undiagnosed for many, many years. And the purpose of this consortium was try to actually provide a diagnosis, which is a very important first step towards understanding why they have that certain disease, and then try to move on from there towards therapeutic research. The undiagnosed patients and that anybody that lives in the US can apply through this program through a website called Gateway that the Coordinating Center at Harvard has generated. And then information about you and your clinical records will be sent to one of the 12 clinical sites that's scattered around in the US, including Baylor is one of these sites here. Patients that get accepted in this program would go to one of these clinical sites and kind of get a thorough phenotyping done at the clinical setting, as well as they'll have their DNA sent for whole exome or whole genome sequencing. And some patients would also undergo metabolomics profiling if that's metabolomic diseases are suspected. This alone would provide about a diagnosis for about 25% of these very difficult to diagnose cases, but many patients would still remain undiagnosed even after using all the state-of-the-art technology that we have at hand. It's not usually it's not that they don't have any candidate genes, it's just that they have some candidate genes, but they don't have really confidence that that's really the cause. For example, you have a missense allele in a gene of interest that has never been associated with disease in human. The clinicians can't really conclude just based on that data, this is the cause of the disease. And one important thing that we can contribute as people that are studying model organism using genetic technology is to perform functional studies of these variants of unknown significance or functions of genes that's never been linked to human diseases. So for these cases, when these clinical sites have a good candidate gene, they'll provide that information together with the brief information about the patient's phenotype to the Model Organism Screening Center, or we call it the MOSC, MOSC. And when we get this information, we don't immediately start doing experiments. The first thing we need to do is a series of informatic analysis, which I'll come back to. And this is the context why we developed MARRVEL. Well, once these informatics analysis is done and we have confidence that this is a good candidate gene, and also we have some ideas of which model organism would be most suited for such study, we'll prioritize these variants for functional studies in either one of these three organisms, C. elegans, Drosophila, and zebrafish. And then we perform in vivo experiments to provide functional data, and together with the clinical data, genetic data, we often end up getting a diagnosis. And our site has been contributing to about 10% of the diagnosis in the entire undiagnosed disease network. So in the UDN, this is what happens. A clinician would give us information about, here's the candidate gene and variant that was identified in this patient. Is this a good candidate? And then what can you do in a model organism? So what we need to do is first gather information about the gene of interest. So what is known about this gene in human? We do literature searches in PubMed. We try to see if this gene has been or has not been associated with a human disease. And there's many other websites that you can go to curate, for example, are mutation in this gene or variants in this gene identified in a control population, or they identified in a disease population? Where is the gene expressed in humans, and so on and so forth. Once we finish gathering this information in human, we also need to see, is this gene conserved in other organisms? We use a tool called DIOP, which is, I'll come back to it, which is very useful in predicting, assigning potential ortholog candidate gene. But in some cases, we need to do blast searches using protein or nucleotide blast. And once we identify that these genes do have a model organism ortholog, we also need to then go into the individual databases like mouse. For mouse, we need to go to MGI, rat, ZFIN, fly-based, and then worm-based. And then for C. elegans, Saccharomyces genome database, PALM-based for yeast. And also we take advantage of multiple of these cross-species data mining tools, which I think later in this GSA's webinar series, Alliance of Genome Resources and Monarch Initiative are going to be coming and giving a talk about their website, which really kind of is complementary to our efforts. So life without MARRVEL. So this is what we used to do. We actually, for each of the genes, we open up these browsers or tabs of different website for human genetics, genomics, ortholog prediction. And then once we gather that information, we go to the model organism, figure out what they do. And then on top of that, we'll have a lot of additional information about the expression of genetic interaction, protein interaction in the literature. And as you can see, this is a lot of information that we need to gather from many different places. And it's also sometimes hard to use these databases. For example, I know how to use Flybase because I'm an expert in, I use fruit flies in my laboratory. However, mining Wormbase is actually very difficult for me because the user interface is very different. The vocabulary is very different. And again, I think everybody has kind of this experience of getting information about their model organism interest is relatively simple, but if you just cross an organism, this becomes very difficult. So this is the context where we reached out to Zhandong's team, who develops programs in addition to his main research interest. And this was in 2017 that we developed this MARRVEL tool. So the idea was that we want to actually have one website that we can go to collect all this information that I just told you about human database and also model organism database. We wanted to use, and so this is what it looks like. So Life with MARRVEL is actually just one page that we have very simple input interface. And then based on that, we get all the information that we would need. So when, while we developed this just for ourselves, we made this tool public because we thought that there was going to be different ways that not only us, but other people can use this website. For example, if you're a clinician and you just had your patient sequence, and then the person didn't get a diagnosis because they didn't have known variants and known genes, but instead she got a list of genes that rare variants that was identified in this patient. So she can use MARRVEL to actually figure out which one of these genes and variants are going to be useful. Or you can come from the completely other direction. If you're a model organism scientist, just like most of you are right here in the audience, if you find something very interesting about your model organism gene X, you may want to know whether the homolog exists in humans, and if it does, whether it's involved in a human disease, or if it's not, what is the possibility that the gene causes a human disease? Upon publication of our paper, we also published several tutorials as in the form of papers or videos, which are listed here. And this has been well accepted, especially in people that are doing clinical work. And that's been since 2017. And ever since we actually developed MARRVEL version 1, we've been improving this by adding new features. And currently MARRVEL is in version 2, which we hope to have another publication about the new features that have been added to this software. For example, we try to add some more flexibility and ways to search MARRVEL. For example, instead of just genetic coordinates that in order to specify specific point mutations, now people can actually use amino acid changes, and I'll show you how that works. Or instead of actually typing in just one gene, that some people may have a list of 50 genes that they want to get information. So you can use you can screen for multiple genes using VCF files, for example. We also integrated additional databases. Again, we developed MARRVEL in 2017, but there's new resources that have been generated over the years. So we want to keep up with all of that. We have prediction of variant pathogenicity integrated now. We also have integrated model organism data from the Alliance of Genome Resources that is displayed in a very comprehensive manner. We also are currently developing, we have a beta version that's up right now, cross-species phenotypic comparison, and also additional information, for example, drugs that work on that specific protein, predicted protein structure, and we're also working on integrating primate genomics data, which is in progress. We also try to improve, based on the feedback we get from the users, improve the user experience by, for example, in the previous versions, different kind of human genetics information was kind of scattered, but we now have a clear division of disease database and control database, for example. Or we have actually, instead of having tables of text, we try to use visually useful ways, for example, ribbon style diagram to show display gene expression, gene ontology, term phenotypes across species. And then finally, we also have been developing several APIs. These are codes that machine can be used to actually query MARRVEL, the data that MARRVEL basically accumulates, so that they can be used for other purposes. And Zhandong is going to be giving you an example of how new software can be developed based on these information. So today I'm going to be switching over to do a little bit of a demo. So because many of you are a model organism researcher, in the first scenario, I want actually going to be starting with a gene that I've been working for a long time, the fly gene Notch, and try to gather information about its clinical relevance in humans, also in other species. And then case 2, this is going to be relatively short because I'm going to be doing most of the discussion of this gene, the program in case 1. But this is a scenario when if somebody comes to you saying that, hey, I have a point mutation in this one gene, for example, this threonine 296 methionine in this gene called GLRA2. What does that mean? Could this be a disease gene for my patients? And what is the functional consequence of this mutation? So now I'm going to be switching over. I hope you still see my screen here.
Yes.
Okay, so this is MARRVEL. Just to remind you, the MARRVEL is actually right here, http://marrvel.org, marrvel.org. Make sure you put 2 R's, because if you don't, it'll actually, Google will probably move you to the comic book store. But here, what you can see is in MARRVEL 2. Note that MARRVEL version 1 is still available if you actually want to use that, but it has been the development of this has not been continued, so I'll recommend everybody to move on to MARRVEL 2. And this is where you can get information about about us, about some questions that you might have. If you want to provide feedback to us, please use this feedback button right here. And then let's see. And I'm just going to be going back. And then if you want to use MARRVEL APIs, if you're a computational biologist, you can also come to here to fetch these codes that you need to gather information from MARRVEL. But today, the main thing that I'm going to be doing is to give you an idea about how MARRVEL would work. So let's say that, again, I'm a model organism researcher. So I start with this 3rd button saying model organism gene. And you can pick your model organism that you work on. I work on flies, so the organism would be fly right here. And then you can type in your gene of interest. Here, you need to use the official gene symbol of your gene your species of interest. And then once you start typing this, it'll actually give you candidates, for example, that starts with that alphabet. But the fly gene Notch is actually a 1-letter capital Notch. So I put that information and then search MARRVEL. The first thing MARRVEL would do is give you a list of ortholog candidates for that gene. And maybe if you already know your ortholog candidates, you can just immediately go to the first tab and just type in that human gene name in the first window. But if you, for example, don't know what your ortholog candidates are, you can start with this. So I hope this is going to be loading. It's taking more time than it used to. Let's see if, here you go. All right, so this is a long list, but just bear with me. So it basically generated a list of genes, human orthologs right here, that starts with Notch 1, Notch 2, Notch 3, Notch 4, which I know are the orthologous genes that I have. There are other genes, for example, Notch 2 NLA, Notch 2 NLC, and Notch 2 NLB, Notch 2 NLR. These actually are paralogs of Notch 2 that has been specifically duplicated in the human lineage. So these can be also considered as orthologous genes to human. And then after that, you see other genes like CRUMS1, CRUMS2, eyes shut. We know that these are not orthologous genes to the fly notch gene. And the reason they're actually shown here is probably because they share the domains, for example, that EGF repeats there's abundant EGF repeats in notch as well as CRUMS and eyes shut. And I think that's why these genes are coming up. And in order to judge whether these are good candidates or not, I want you to pay attention to this DIOPT score right here. So DIOPT is an ortholog prediction program. And for example, between human and flies, there's 16 programs algorithms that DIOPT uses to predict whether a gene is a good ortholog candidate or not. And here you can see that the score of notch 1 is 12 out of 16, which means that 12 programs out of 16 algorithm basically predicted that this is likely to be an ortholog. And that is on the top. For notch 2, you can see the score is 10, notch 3 is 10, and the notch 4 is 5. So these are not as good as the first one, but it's because of the sequence divergence between notch 1, notch 3, and notch 4, for example, are much more larger than between notch 1. And one thing that you can also look at is this best score from human gene to fly. So this is a case where the diap score here is actually shown as, okay, when we put in the fly notch, notch 1 comes up with a diap score of 12, and that's at the top of your list. What if we do the other way around? If you put in notch 1, does the fly gene notch come to the top of your list? And if it says yes, it's likely that these are good candidates as well. So you can see for notch 1, 2, 3, 4, even for the notch 2 and LA, this also says yes under here. Note that, under this, you can see for crumbs, you can get a score of 2 out of 16. So that's, again, 2 programs predicted that this is an ortholog candidate, but you can see that it says no right here from the human gene to the fly gene. Notch is not at the top of the list because there's another gene called crumbs, which is actually the orthologs of crumbs 1. So this is how you can kind of go through. And then genes that are listed with a diap score of 1, I typically ignore it because these are, again, just distant homologs or genes that actually share some kind of a homology between your gene of interest. So out of this, we can go to any one of them, but why don't we go to notch 1? Say that I don't know anything about human notch 1, tell me about human notch 1. So you can, in order to do that, you actually click this MARRVEL link and then just say MARRVEL it. And I hope the loading would not take that long. Oh, here we go. This is what a MARRVEL search results would look like for notch 1. And by the way, you can actually, if you want to come here directly, you can go back to the MARRVEL top site. And if you already, again, know that notch 1 is an ortholog candidate. Let's see if this is working. Type in a little bit slowly. There you go. If you hit search just with putting in the human notch 1, it should basically take you to the same window. So what do we see here? This is actually MARRVEL is, again, it's a 1 page, so you can kind of scroll from all the way from top to bottom. You can see that there's a lot of information. Don't freak out. But again, this is the information that MARRVEL basically gathers from multiple different databases and then puts it into a single HTML file for you. So why don't we go back to the top of this window and tell you a little bit about what information I typically try to extract from here when I'm looking at this. So on the top, it would give you the human gene name. It actually has multiple links to other database, which if you want to actually gather more information about this, it would have the name. It's called Notch receptor 1 in humans. And if it has alternative names, for example, Notch 1 has been called AOS5 in some papers, or AOVD1 or TAN1, you can actually see it here too. It gives you first a summary of Notch 1 from the gene entry from NCBI. So it tells you that this gene encodes a member of the Notch family proteins, member of this type 1 family, and blah, blah, blah. So especially, I know about Notch 1, but if there's a gene that you're searching that you have no idea about it, just reading this might give you an idea of what that gene looks like. And then you can see on the right side, you can also use this actually navigation bar right here. And you can see that the information that's shown in MARRVEL is actually split into kind of different categories. For example, disease databases, control databases, model organism, drugs and ligands, and proteins. So what we're starting here is actually start of the disease information here that actually starts with a database called Omen. Omen stands for Online Mendelian Inheritance of Men. It's kind of like an encyclopedia of genes and humans that's linked to Mendelian disorders. So here you can have a little bit of, you can just read the summary. There's 2 phenotypes. Typically this phenotype means 2 diseases that are associated with Notch 1, and there's 12 reported alleles that are shown in Omen. So again, it would give you kind of the gene name and then a short description from Omen about what the gene is. And here's a table of the two phenotypes that are associated with Notch1. One is called Adams-Oliver syndrome 5. The other one is called aortic valve disease 1. It actually shows that both are autosomal dominant conditions, and then it actually has links to the specific disease database. So if you click on this, it would actually send you to the Omen page about Adams-Oliver syndrome, which you can read more about what this condition is. If you're interested in what the phenotypes this patient has, you can click on this clinical synopsis and it will actually tell you about the disease condition. Again, MARRVEL would always have links out to the original databases so you can actually read more about it. So now I know that Notch1 is involved in a human disease, and there's actually two diseases that are autosomal dominant. And here is followed by a list of alleles. For example, this is an 85 kilobase deletion that causes Adams-Oliver 5. For example, the third one is a cysteine 429 to arginine missense mutation that causes Adams-Oliver syndrome, and so on and so forth. So we can get an idea of maybe a handful of genes variants that's reported here. Although I just need to make sure that, mention that Omen is not a comprehensive, it's actually kind of an encyclopedia. So this is not a comprehensive list of all the variants associated with this gene. That's why we have also other databases. For example, ClinVar. ClinVar is a disease database. It's typically clinicians that identified variants in these genes in patients would actually report this in ClinVar. And for here, there actually is 97 pathogenic variants that's associated with Notch. And then there's also 36 additional likely pathogenic alleles. And if you want to see the alleles, you can kind of open this up and see what they look like. Genome2MP and also Decipher or other disease databases that would actually, these are coming mostly from undiagnosed disease patients, and they'll basically give you a list of variants that were identified in Notch1. So if you want to actually identify patients, and also if you want to connect with clinician that sees these patients, information from here could be useful. And again, you can open up and see what the variants are to see what kind of variants are identified in patients. And then we go on to a series of what's called control database. Control database are typically sequence data from healthy people. And the most important one we use is called GNOMAD, GNOMAD. This is a database of about 120,000 whole exome sequencing in addition to 15,000 whole genome sequencing that's performed on relatively healthy individuals. Definitely it's excluding severe pediatric conditions, but it might actually have people's information sequencing data from patients with like a neurodegenerative disorder, late onset, or maybe people with psychiatric conditions. But at least you can actually use this if you say, let's say your patient of interest is a pediatric patient and you want to see if a specific allele is found in another healthy people or is kind of excluded. In addition to variant level information, you can get gene level information. And this is showing you a table of synonymous variants, missense variants, and loss of function variants, meaning stop codons or frame shift or splice variants that are identified in Notch. There's two numbers, expected and observed. So what this is for a gene size like Notch, for example, you expect to see 108 stop codons or a loss of function mutations in the gene called Notch. And then in the actual data, you only see 5 patients, 5 individuals with stop codons, which suggests that there seems to be some kind of a selection that's going on in the general population against loss of function alleles. And this can be also visualized by this observed over expected ratio, so 0.05, or this number called the PLI score, which is a score between 0 and 1. 1 means that there's a selection going on. So this actually already tells you that loss of function allele of this gene is not correlated, versus for Notch4, if I looked at the same data, the PLI score is 0, for example. There's many people with loss of function variants walking around in the population. And then in addition, we have control data for copy number variations from GDV and also copy number variation from Decipher. So these two databases are not only the stop codons or missense variants, these are kind of large or small deletions that are identified. So here you can kind of use it as a way to rule out or rule in whether my gene of interest, haploinsufficiency of my gene of interest, for example, could be associated with disease. So we're done with all the human related databases. We're moving on to model organism data. So here, then we can have, we have a table of a list of ortholog candidates that DIOP predicted, not only in flies and human, but also in different animals between mouse, rat, zebrafish, fly, worm, and budding yeast. Here you have the links to individual specific website, and then it will also give you the DIOP score, and then whether the reverse score, for example, again, from mouse to human, human to mouse, is actually the yes or yes. It also gives you kind of a confidence score that DIOP provides. So here, now we know that mouse is human notch 1 is orthologous to mouse notch 1, rat notch 1, zebrafish notch 1b. For people that knows in zebrafish, about 25% of the genes are duplicated. So notch 1b is kind of seems to be higher than notch 1a. If you want to actually look at additional ortholog candidates, you can actually click on this button so that you can see what other genes are actually shown. But here's a kind of simple table of the top candidate genes. So in flies, it's called notch, and worms is called lin12. Here again, in yeast, there's two genes, ADA2 and GAL4 is actually coming up. But again, you need to be aware that this has only a DIOP score of 1 because there seems to be some homology between the GAL4 protein and the human notch gene. So here you can see that this gene is conserved in maybe metazoan, maybe not in yeast, because we know that these are probably not orthologs. And then this is followed by expression data. So human expression data is coming from a database called GTEx, which is a transcriptomic RNA-seq database. So you can see that there are, this notch gene is kind of widely expressed in different tissues. And you can also kind of click on or click off certain tissues if you want to actually eliminate or kind of zoom into one of these specific organ systems. Also, expression pattern in model organism, this is provided by the AGR. We're going to be showing in these ribbon-like diagrams. So you can see, again, mouse, rat, zebrafish, flies, C. elegans, and the E species. And here also the mouse, you can see that it actually is widely expressed in many tissues. Note that not having the information doesn't mean that they're not expressed often. It may mean that they don't have the data for it. But so here you can kind of get an idea that this gene is ubiquitously expressed in multiple species. This is followed by a phenotype tab that we're still kind of working on. So it's still a beta version, but this would give you different kind of phenotypes that are associated in different species. So if you can kind of hover over these icons, you can see this is developmental phenotype, growth phenotype, brain phenotype, nervous system phenotype, eye and ear, and so on and so forth. And you can see that whenever you see this teal color, there seems to be a phenotype that's associated. If you want to kind of look into what exactly that phenotype is, I clicked on that human neurological symptoms. It gives you kind of these HPO terms, or in the flies, for example, this is the neurological neuron-related phenotypic terms that actually are coming up in these species. So this kind of, again, gives you an idea of what kind of phenotypes that's been linked to this gene in different species. We also have gene ontology registration from multiple model organism that's classified based on the 3 large gene ontology categories. So again, you can get an idea about their biological function by looking at this. And then 3 last thing is that we have Pharos, which is a kind of a drug database that's supported by the NIH Common Fund, which is kind of a comprehensive library encyclopedia against FDA-approved drugs. So for Notch 1, it looks like there's no FDA-approved drugs, but there are several ligands that has been known to be binding to Notch. So in this case, you can actually go to okay, well, what is this? You can kind of go to Pharos and see what basically that is. It'll actually tell you. Yes.
You're going to have to start wrapping up.
I will. To about these chemicals, and then you can actually go to a specific web publication that's associated. Finally, we have protein domains that are associated with this Notch protein, and we also have multiple protein alignment, which you can kind of use to also highlight different regions. For example, I'm going to be highlighting 50 to 100 amino acids. You can, and especially home into specific domains. And finally, we also just integrated predicted protein structure, which you can kind of move around for this is from AlphaFold that's developed based on Google's artificial intelligence, and then specifically go to amino acid of interest to get in there. Yeah, so for just for the sake of time, if you want to actually go into, for example, human protein, you can type in the specific protein change of interest and go here. And if you do that, you will actually convert it to a genomic coordinates, and then you can go to the specific web page for MARRVEL that would actually give you not only gene level information, but about variant level information. For example, whether that variant of interest is predicted to be damaging or tolerant or not. So I'll stop here basically, and then I'll bring it back to the presentation. And using this data, basically, as you're saying, you can, MARRVEL allows you to gather a lot of information, but you still need to have some domain-specific knowledge to make use of this. So we started thinking about, can we automate this variant prioritization process using kind of state-of-the-art computational technology? And that's what Zhandong is going to be telling you about for the MARRVEL AI project. So I'm going to be.
When we first approached this problem, and we interviewed a lot of clinical geneticists, and we were trying to understand their thinking process. And here is a summary. After interviewing 5 to 10 clinical geneticists, they always start with an initial list of candidates, and through various lines of evidence, they can narrow it down. Their goal is to narrow it down to a handful of candidates that are most likely or most probably causal. And how do we computerize this? Our goal is to train the computers to do this. Of course, you have learned a lot of this in the media about our face recognition, how to automatically identify an object from a video, right? All this self-autonomous driving. So training a machine learning and artificial intelligence is something you commonly see. How do we do it in the context of a personal genome and disease diagnosis? So this is basically the layout, the blueprint of our MARRVEL AI. The input of the system is a personal genome along with the EHR, the electronic health record, describing the phenotypes of the patient. And once this is given to the machine learning engine, it has two approaches. The first approach is like this autonomous driving and face recognition. You use deep neural networks that requires a lot of data training data points to let the computer identify something or learn some rules. Oftentimes, these rules are a black box. We can't really understand how they made decision, but the decision is right. Here, we don't have the luxury to have millions or thousands, tens of thousands of data points because we're dealing with rare diseases and undiagnosed diseases. What we decided is to use a hybrid approach to combine our knowledge of genetics with machine learning technologies. Exactly how? So the training data we were lucky enough to obtain is about 500 cases from Baylor Genetics, and that's divided into 396 training and 99 validation set. We also have reserved an independent testing set to evaluate how well the artificial intelligence works. That's 126 samples from Baylor Genetics and 88 samples from the UDNs. For any given sample, we're dealing with on the order of 10,000 variants that are, many of them are common. We're not talking about the rare variants. We're just talking about the file size. Any particular input, the distribution of number of variants can range from 10,000 to 20,000 to 40,000. And many of these cases have either one diagnosis, meaning that one variant is a true causal variant, and a very small portion of them have more than 3 variants being causal variants. Now we know that the cohort we're dealing with, how are we going to develop a hybrid system? So we developed 6 different modules based on biology and based on what Shinya described for different sections of MARRVEL. Some of them is about the human genome sequence database, like module 1. Some of them is about the allele frequency and conservation. Some of them is about from existing tools like CAD review. Some of them you may be familiar with, like Polyfin, right? And if you zoom into one of the modules, for example, here we showed module 1, it is a lot of decision grams. If this, then yes. If not, then go to the other route. So this is a lot of decision trees to tell you on any particular path, given a series of yes, no, yes, and no, what is a probability that you're going to have a fuzzy estimate of the importance of that variance, high, medium, low. A lot of this is just feature engineering. You don't have to make it accurate. That's why it's really fuzzy, right? It's probably high, probably medium. You really don't have to have very exact estimate. So after that, once we have all these modules, we can actually train something called a random forest. You can think of this as a decision tree, but with many, many of these trees. Each tree has a decision node. For example, if the allele has a high frequency in a healthy population, probably not important. If the allele has a low frequency, it's probably we should go on and look for other evidence, right? So these trees, each of these individual tree have 1 vote. And at the end of the day, we do a majority vote. Let's say if you 100 trees or 200 trees, we summarize all these votes together, and then the final yes or no or final ranking is the summary of all these trees. We're not the first group to do this. Actually, in the literature, there are several tools that does similar things as ours, but do it slightly differently. Some of them use artificial data, simulated data to do this. Some of them rely on different set of features than us, right? Not the same kind of a feature engineering. So we compared it to Axomizer, one of the methods in this field, on these 126 independent testing data. As you can see, if we measure the performance by how many times we can rank the causal variance as the top 1, how many times we can rank the causal variance in top 2, in top 5. So here, about 68% of the time, we can rank the causal variance in number 1, compared to 47 in Axomizer in the Baylor genetics data. In the UDN data, the undiagnosed network, we have a 60% of a chance that can rank the causal variance in top 1 and 36 in contrast to 36% in Axomizer. Again, I want to put a disclaimer here that we didn't exhaust fully optimize and find all the combination of Axomizer's tuning parameter. We tried our best. So we're going to keep optimizing it to make it a really, really fair comparison, but this is what we can achieve so far. Right, we did a lot of feature engineering, but the first question we ask ourselves when we get the result is, does the feature engineering work? So we look at all the features and rank them by their importance. There are a way called the Genie Index for those of you who are familiar with the random forest. For those of you who are not, you can think of this as an index kind of measuring how much information condensed is obtained at each node. It was able to identify diffuse Frank score as a phenotype score using string database is important. Like nomad allele frequency is also important. Omen information is also important. So it identified those 6 features to be important. Then we ask ourselves, if we eliminate those features from the trees, from the machines, do I still get a similar result? And to our surprise, if you just eliminated a single one of them, one at a time, the performance don't drop much. original performance is 92. If you eliminate any single one, the performance almost don't change. However, if you eliminate all of them, all 6 of them together, the performance really drops from 92 to 83%. If you do not do any feature engineering, only use the raw features, then your performance is only 78%. 78% the chance you can rank the causal variants in the top 20. So this kind of reverse engineering tell us that, okay, the feature engineering seems to work to some extent in increased accuracy. The next set of questions we had is, well, these trees are artificial trees. Will they learn some sort of biology, right? Will they mimic the thinking process of the clinicians? For example, will they learn autosomal dominance and recessive X-linked? This inheritance mode is important in making the decisions. We look at some of these trees. Indeed, they were able to learn. So on the top is a tree that has a lot of parameters. Below is a simplified version of it. You can see the inheritance first node is it curated in Omen or not? Do I have a similar phenotype in the human genome medical HGMD database? Is it likely to be autosomal recessive? Is it likely to be autosomal dominant, right? Based on the decision, yes, no, true, false, it's going to reach the decision of causal or not. Some of the trees indeed can learn that how to differentiate these autosomal dominant or recessive mode. How does it handle known diseases that are curated in Omen versus those unknown diseases that requires novel variants? So we first look at the known diseases. Take the example here. This patient has neurological features like delayed speech development, seizures, and also has dysmorphic features like abnormality of the face. So we took this patient's genome, run it through the random forest. On this graph, each dot is a variant. The y-axis is a predicted score, and we ranked it from position top 1 to top 40. As you can see, the top 1 has a really high rank compared to the number 2 and number 3, right? It did really see a drop of the score. And then there, in position 5 and 11, there are 2 more variants that are also ranked important, and according to the truth, the clinical annotation, they're also important as well. It turns out that this is a kind of a mixture. The first gene, ATP, causes alternating hemiplasia. If you look at the features, the phenotypes, it kind of partially matches with the patient's phenotype. And those ranked the position 5 and 10 actually is a recessive feature, that recessive mutation that causes conical tooth hypotonia that partially matches with dysmorphic features. And indeed, it can kind of identify variants that are curated in Omen. What about this truly hard case that has never been discussed, never been discovered before? This is kind of the ultimate test. We tried several of these. And one of the examples I want to show here is one patient that has a global developmental delay, epilepsy, and then some more detailed phenotypes of seizures and intellectual problems. We give this HPO and the genome into the random forest. You can see that there are the causal variants that we predicted is actually also the causal variants predicted by the human expert. And compared to some already curated variants that cause known disease, there's a huge difference in terms of a score. There are 2 main lines of evidence that we believe that machine reached the decision. The first line is it's low frequency. Although it's not curated in Omen, but it has a very high GERB score. It's likely pathogenic based on ClinVar and also not autosomal recessive and has a high PLI score. The second line is also focused on the gene function. It has a very low frequency in the loss of function mutation in NOMAD, right? It's basically based on the type of mutation that we're dealing with. And this case is solved because of multiple evidence. First of all, people look at a larger database independent UDN from other databases, they see other cases like this. Case 7, 8, 9, 10, 11, they all have the same mutation or same mutation. 7, 8, 9 have the same mutation. 10 and 11 have a slightly different mutation, but a very close position. Same amino acid. And when you knock out this protein, TRIM8, in the model organism, you see like some functional importance of this protein, the nuclear bodies, localization changes. So to summarize, we develop a MARRVEL AI systems that can reach a decent diagnosis accuracy for both known diseases and unknown diseases. And the MARRVEL system provided a very fast data wrangling. One lesson we learned is actually to make these data machine learnable. We spend a lot of the time trying to wrangle many databases information into computable format. And MARRVEL AI API did a great job in streamlining that. And our hybrid system enabled us to peek into how the machines make these decisions. And also kind of we have other examples where machines made very obvious errors, and the human experts can help us correct for it. And with that, I'll give it back to Shinya.
Okay, so this is the final slide to wrap up. So I hope we gave you an idea of what the MARRVEL project was, where it's coming from, where it's now, and what it can be used for. For the MARRVEL original search engine that we developed out of our need has now evolved to MARRVEL 2, and we'll keep on adding new features to it so that we'll basically be able to collect a lot of information from different web sources and put it into one place. As you saw in Zhandong's talk, this putting in one place in a harmonized manner is very important, because now not only biologists, but computational bioinformatician can start using that in a creative way. One application was to take variants identified in patients and predict which ones could be pathogenic. And eventually we're going to be having MARRVEL AI up running so that clinician can basically utilize that to support their clinical activities. And finally, in parallel, we're developing this website called Model Matcher, and this is a website to connect clinicians and scientists who share interest in the same gene. Last month, Michael Wangler basically talked about that, and the idea right now is that if we can integrate all this basic biological knowledge with machine learning based knowledge, together with expertise and clinicians and scientists, we hope to advance this rare and undiagnosed disease field. So in summary, this is there's a lot of people involved, but I just want to give especially a shout out to Julia Wang, who was an MD PhD student in Hugo Ballen's lab, for initiating the MARRVEL version 1, and there's people that are continuing on to develop this. And then Don Shui Mao, who's a postdoc in Zhandong's lab, has been spearheading the MARRVEL AI team. So with that, I'd like to thank everybody, and then take any questions that people might have.
All right, thank you very much, Shinya and Zhandong. I suggest that people who want to ask a question through the internet can raise their hand. I'm not sure that will work, but one thing that will certainly work is if they go to the chat function and post their questions in chat. So there were already a few questions in chat, and that is, how do you identify the animal that is suitable to your study?
Yeah, so maybe I'll answer that. So it kind of, again, usually in the UDN, we need to select between C. elegans, flies, and zebrafish. And obviously, if you know, you can actually always consider mouse and other species. For the UDN, what's important is that we don't have unlimited time and unlimited money. So if we can actually do a functional assay in a more simpler organism, we prefer that. So for example, if we can do an experiment to show that a mutation or variant of interest is a loss of function or a gain of function, and we can do it in C. elegans, worms, flies, and zebrafish, we typically assign it to the worm core. But sometimes the gene is not conserved or the amino acid is not conserved. We basically go to animals that basically requires more time. But this also has to do with maybe the phenotypes, the kind of phenotypes that are seen in the patient. If it's, for example, epilepsy, we can actually study it in a fly because there's an assay system called bank sensitivities. There's a lot of parallels between it. And but for like, immune system function, especially if it's related to T cells, we can't study in flies, so zebrafish needs to do. So I think that this is kind of a case-by-case scenario, and it's always good to discuss with expertise, and especially clinicians, what is the question and what is the answer that you need.
All right. So there is another comment from Christopher Jones. Differentiating between not expressed and no data available would be a useful improvement. And he said, set by someone with absolutely zero clue whether it's possible to extract that from the relevant databases.
Yeah, I think so. Yeah, I think that's a great suggestion, and probably we'll definitely look into it. Again, for like GLRA2, let's go to here to the expression. You can again going down to here. This expression here is actually, for the humans, you can see it is actually expressed in the nervous system, but not in other species, other places. So this is obviously positive data here. For here, I guess I'm seeing the expression pattern here, and again we see in the nervous system and the mouse, we see nervous system in zebrafish, flies, C. elegans. But no data, probably, again, nothing at all in the rat. And if you actually want to go to the original data to see if the rat gene has been done or not, you can click on this. It would go to get you to RGD and try to figure out why we're maybe lacking that data. That's a great suggestion. If we can actually distinguish between, for example, if it's not detected, we can maybe gray it out in a different color or something. And we are actually doing that for the phenotype, for example. If this is not mentioned or we don't have data, it's actually grayed out in a darker color. But if it's the data is present, it's negative, it's in this lighter gray color here.
All right. Any more questions? Concerns? I see no other postings. If you have a question, you're more than welcome to email to Zhandong or Shinya, and I'll.
And again, people can always use the feedback, especially if you have a specific question about MARRVEL. You can use this feedback button and then type in your name and email, and we'll try to get back to you.
All right. Thank you very much.
Okay, great. Thank you. I hope this was useful.
We'll have a next one in about a month, and we'll advertise it to GSA as well. This one will be related to AGR and other databases. So there's a few more talks that will follow. Thank you.